Good day everyone, I am Angel Sorayas from BSN1G. For this activity, I will demonstrate and explain the process of RNA translation through a simple simulation. But first, let us know what is RNA translation. So, RNA translation, it is the process by which the information carried by the messenger RNA or also known as mRNA, it is used to produce a protein. In simple words, parang may instructions or recipe ang mRNA and the ribosome reads the instruction to build the protein using the amino acid. For my simulation, I will use a simple analogy. mRNA is the recipe. The ribosome is the machine that reads the recipe. The tRNA is the delivery worker and the amino acids are the ingredients. Now that I'll give you some knowledge or brief introduction about RNA, let's talk about codon and anticodon. A codon is a sequence of 3 nucleotides found on the mRNA. It contains the instruction for which amino acid should added to the growing protein chain. On the other hand, an anticodon is a sequence of 3 nucleotides found in the tRNA. It pairs with a codon on the mRNA through a complementary base pairing. For example, here we have the mRNA codon, which is AUG. Its complementary tRNA anticodons is UAC. Remember that in RNA, A pairs with U while G pairs with C. Notice also that mRNA is read in the top 5' to 3' direction while the tRNA anticodons pair in the opposite 3' to 5' direction. So in simple terms, the codon is the instruction of the mRNA while the anticodon is the magic sequence of the tRNA that helps bring the correct amino acid. This matching process allows the ribosome to build the correct protein. Now let's proceed to the first process, which is the initiation or beginning of translation. Here we have our mRNA. The mRNA contains a group of 3 nucleotides called codons. Each codon gives instruction for a specific amino acid. The mRNA enters and attaches to the ribosome. The ribosome then looks for the start 2 codon, which is usually AUG. AUG is important because it signals the ribosome where translation should begin. It also codes for the amino acid methionine. Now, a tRNA carrying the correct amino acid comes in. The tRNA has an anticodon that matches the codon on the mRNA. For example, if the mRNA codon is AUG, the tRNA anticodon is UAC. So, initiation basically prepares the mRNA and the ribosome to begin building the protein. Now that you already know the first process, let's now proceed to the second process, which is elongation. This is the stage where the protein chain starts getting longer. So the ribosome moves along the mRNA and reads one codon at a time. As each codon is read, a tRNA brings the correct amino acid. So for example, if the next mRNA codon is UUU, the matching tRNA has the anticodon AAA. This tRNA brings its corresponding amino acid. The amino acid are then connected by peptide bands. I will now connect these amino acids together to present the growing peptide band chain. The ribosome continues moving along the mRNA, and more tRNAs bring more amino acids. So the process is basically codon is read, matching tRNA arrives, amino acid is added, peptide band forms, protein chains gets longer. This continues until the ribosome reaches a stop chain. Finally, we have termination, which means the end of translation. When the ribosome reaches a stop codon, such as UAA, UAG, or UGA, There is no tRNA carrying an amino acid that matches the stop codon. Instead, this signals the ribosome to stop translation. The newly formed polypeptide chain is released from the ribosome. After that, polypeptide chain can fold into a specific three-dimensional shape to become a functional protein. I also include the application to nursing. Why is RNA translation important? RNA translation is important because proteins are essential for many processes in our body. Proteins can function as enzyme, hormones, antibody receptors, and structural components of cell. They are also important for growth, tissue repair, metabolism, and normal cell function. As future nurses, understanding protein synthesis helps us understand how cell normally function and how problem at the molecular level can contribute to diseases. For example, if there is a change or mutation in genetic information, it can sometimes affect the protein that is produced and may affect normal body function. Therefore, RNA translation is important because it connects genetic information to the actual proteins that our body cells need to function. To summarize our study, RNA translation has three major stages. First, initiation. The mRNA attaches to the ribosome and translations begins at the start codon. Second is elongation. tRNAs bring amino acids, and the amino acids are connected through peptide bonds to form a growing polypeptide chain. Third is termination. The ribosome reaches the stop codon, translation ends, and the newly formed polypeptide is released. So remember, mRNA gives the instruction, the ribosome reads the instruction, tRNA brings the amino acid, and the amino acid are assembled to make the protein. That is the process of RNA translation. Thank you for watching.